All right, so the last process of membrane transport that we want to talk about is bulk transport. It is a type of active transport in the sense that it takes energy, but it's bulk transport is different in the sense that we are moving large amounts of material into or out of the cell all at one time. Okay, so think of like active transport, passive transport. It's kind of like going to the local convenience store. You just kind of need like a gallon of milk. You may not need to do like a whole full-on grocery shopping trip, right? But if it's been a while since you've been shopping and you need to stock back up on a lot of stuff, you're going to head to Costco and you're going to get stuff in bulk, right? That's kind of what's happening here with bulk transport. Not really, but maybe that analogy helps you remember the difference, right? Bulk transport, moving lots and lots and lots of stuff all at one time. So bulk transport is how cells eat and drink, right? So this is an image right here. We're going to use this kind of as an example. This is an immune cell called a macrophage. Macro meaning large, phage meaning eater, right? Or to eat, okay? So these are cells in our immune system that find and destroy bacteria by eating them, okay? But how do they do that? How does the cell eat? It doesn't have a mouth, it doesn't have a stomach, right? How does the cell eat, okay? This is what it looks like. I want to play this real quick. So this is our macrophage right here. This is all of the bacteria. Watch it go. It's engulfing and literally just wrapping its body around those bacteria. And it's going to go for the other group over there too. Look at that. It just ate all of those bacteria. How crazy is that? Right? So how does this work? What's going on here? That's kind of our question here. We're going to use this as our example of how how bulk transport works. All right, so again, the key here, watch what's happening to its membrane. It's macrophage's membrane right here. It's extending it around that group of bacteria. Okay, that's basically how bulk transport works. So, we've kind of talked about this process before, but we didn't call it bulk transport. We've talked about molecules moving out of the cell secreting molecules using vesicles, right? So something is produced in the reservoir, gets transported to the Golgi to be modified and folded properly and be attached to some carbohydrates, whatever. Then the Golgi can secrete that product out to the environment in a vesicle, right? That vesicle, that membrane bubble, will just fuse with that membrane, okay? That is a type of bulk transport, okay? So we can secrete large amounts of material out to the environment in a vesicle. It's a type of bulk transport. And we can do the reverse process as well. We can take molecules in to the cell using bulk transport as well. Okay, and using these vesicles as well. That's basically just what's happening is we're exchanging vesicles with the plasma membrane to either bring material in or take material out of the cell. So we give it different names depending on what direction the material is moving. Exocytosis, remember cyto means cell, exo means outside. We are moving molecules or material or waste or signaling molecules, whatever it is that the cell needs to get out of itself. Exocytosis is the process of a vesicle containing either waste or maybe it's a hormone or a signaling molecule or whatever. Vesicle containing that stuff it's going to get rid of moves to the cell surface and moves along the cytoskeleton to the surface of the plasma membrane and then fuses and merges with that plasma membrane, and in the process all of that stuff that was in that vesicle is now outside the cell. It's in the extracellular environment, in that interstitial fluid, that's between the cells. Okay, so like in the process of maintaining our constant blood sugar level, right? Blood glucose levels spike up. Let's say we had a really nice sugary breakfast or something, right? Our blood sugars spike when we consume all that sugar. Our pancreas is like, whoa, slow down there. That's way too much blood sugar. We need to get that back into normal levels. So it pumps out a bunch of insulin. So it goes through this process of putting a bunch of insulin, a protein, into a vesicle. That vesicle fuses with the surface of the pancreatic cells, which makes it into the extracellular fluid, or the interstitial fluid, the fluid between all of our cells, which is eventually picked up by the circulatory system and moves to the rest of our body. And the rest of our cells, when they come into contact with insulin, they're like, oh, we should probably grab some of this extra glucose. And they bring it into their cells through usually active transport or facilitated transport. Okay, so exocytosis, outside cell. Vesicles move to the surface of the cell, merge, and then anything that was inside of that vesicle gets distributed to the outside environment. Now we're just going to reverse that process. Endocytosis. Endocytosis is inside cell, basically, right? Endo means inside, cyto meaning cell. So cells can use vesicles, just like that vesicle that fused with the surface, they can use a vesicle to bring stuff into the cell as well, right? So this is how cells eat and drink. So we don't really think about cells drinking, but they do. They might need to take in a large quantity of solutes all at one time. So we kind of break up endocytosis into two types: phagocytosis and pinocytosis. Phagocytosis, so that prefix phage, means eating. Cyto meaning cell. So cell eating is what phagocytosis means. So in this process, we're going to take the membrane, just like that macrophage that we saw, that membrane is going to extend around a food item or whatever it wants to eat. It's going to extend its membrane around, trap it, fuse it on either side, and then move it inside using a vesicle. Okay, so phagocytosis is a process of extending membranes around a food item, trapping it in a vesicle, and then that vesicle moves inside the cell with the help of the cytoskeleton. Pinocytosis is cell drinking, basically, right? So if there's a concentration of solutes on the outside of the cell, the cell's like, ooh, these are good solutes, I need these right now. Maybe I need some extra sodium or I need some extra potassium, whatever it is. If the cell needs to bring in a large amount of solutes, it can do the same thing. But it's not going to trap it around food molecules, it's going to just trap it around whatever molecules are there, whatever solute molecules are there, and then boop, bring it into the cell. Okay, same idea. But the difference is we're bringing in basically just solution instead of food. So the question is, though, hopefully this question popped in your mind, but like, how does this cell know to eat the bacteria? How does this macrophage know to eat these bacteria, right? How can it tell that it's a bacteria? Well, it's a form of what we call receptor-mediated phagocytosis. Okay, so it's not just going to go around eating everything. If it did that, it would eat all of our cells, and that would just be bad, right? It only needs to eat specific cells. We only want this macrophage to eat specific cells. We really only want it to eat the bad bacteria that our immune system's trying to get rid of, right? We don't want our macrophage going and eating all of our symbiotic bacteria, right? So it uses a process of what we call receptor-mediated phagocytosis, meaning that that process of phagocytosis will only be triggered once receptors on the outside of that cell are activated. Once something binds to those receptors on the outside of the cell, that then begins the process of phagocytosis or pinocytosis. Okay. So solutes, in this example, we're looking at receptor-mediated pinocytosis. So in certain areas of the membrane, there'll be receptors that once they are bound to a specific solute, they will then trigger through that cell signaling process, cell signal transduction, they will trigger that process of creating that vesicle and bringing in those solutes. Okay, so the only difference here is that this process will only begin once the receptors are activated by binding to their intended substrate. So the receptors, what type of molecule do you think they would be? They're going to be proteins. They're going to be enzymes probably, right? So they're going to bind to their substrate. That then triggers a signal that's sent to the rest of the cell. Cytoskeleton kicks into gear, creates that vesicle, right? So the process of creating that vesicle, that's all done by the cytoskeleton. helping that plasma membrane move around. Remember, because the cytoskeleton is anchoring into that plasma membrane. So vesicle formation is a process that is carried out by that cytoskeleton. But how does this work for macrophages? What is their receptor that they're looking for? So macrophages on their cell surface, so they have little extensions of their body. They're super cool, but they have extensions of their body that are coated with these specific receptors that we call Fc receptors. And basically what these receptors are looking for are antibodies. They're really looking for an antibody-coated bacterium. So antibodies are those proteins, those defensive proteins that our immune system makes, that they will bind to the surface of pathogens, bacteria or viruses, right? And they basically neutralize that bacteria or virus. They keep it from dividing. Antibodies keep that bacteria from dividing. Antibodies keep that virus from importing itself into our cells. But just because it's caught up with the antibodies right there, we still need to get rid of it, right? So macrophages are kind of like the cleanup crew. Once the pathogen, bacteria, virus has been neutralized by these antibodies, we now got to destroy it, right? So macrophage, their cell membrane receptors are looking for these antibodies that have been bound to the surface of a bacterium or a virus. And then once their receptors on their cell surface bind to some of those antibodies, that then triggers the process of phagocytosis. Okay, so receptor-mediated phagocytosis, we wait for receptors on the cell surface to bind to their substrate, and then that activates the cytoskeleton to begin the process of creating that vesicle to bring that material into the cell. Then macrophages, all right, cool. I just ate a bunch of bacteria. Yummy, yummy, yummy. What I'm going to do? Lysosomes will fuse with that phagocytosis vesicle that I just brought in. Lysosomes, remember lysosomes are the organelle that are vesicles that are full of digestive enzymes, right? So lysosomes will fuse with that vesicle that macrophage just used to surround the bacteria. The lysosomes dump their digestive enzymes into that vesicle, which breaks down the bacteria. Okay, so that's how our cells basically eat. That's how other cells eat smaller things. Cytoskeleton triggers vesicle. Vesicle gets brought in. Lysosomes come in, bring in their digestive enzymes, and break down that whatever food molecule or other cell was just consumed. Macrophages do a lot of other cool things beside that, but that's a teaser for once you get to the immune system. But they're going to take pieces of that bacteria that they just consumed and put them on the outside of their cell to trigger other immune cells. You don't need to know that, it's just really cool. So they basically take pieces of their dead foe and attach them to the outside of their cell membrane and say, other immune cells, look at what I just caught. We need to kill this. Okay, sorry. Got a little ahead of myself there.